Friends, welcome to Biology Made Easy. Today in this video, we are going to discuss the microbodies. So from the very name you can guess microbodies, that means they are very small in size and they are surrounded by a single membrane. I will discuss about microbodies. So microbodies are very small in size and usually they range from 0.5 to 1.7 micrometer. They are so small in size and they are surrounded by a single unit membrane, not two membranes like that of the chloroplast and mitochondria. So though they are very simple in structures, but their function is diverse. That we say they perform different types of functions, and these microbodies are present in plants, plant cells, animal cells, fungi, and also some protists. That means these are present in plants, animals, fungi, and protists. The microbodies are mainly of two types. One is peroxisome and the other is glyoxysome. So there are two types. One is peroxisome and the other is glyoxysome. These two are known as the microbodies. So let us discuss first this peroxisome. So the peroxisomes were first discovered by Christian D. Duvet in 1965 from the liver cells. And these are found both in case of plants and animal cells, found both in plants and animals. Now, why they are known as peroxisome? What is the reason behind this? It is because they both that is the peroxisomes, they generate and they destroy hydrogen peroxide. That means they give rise to hydrogen peroxide and also they break it. Suppose let us take one example. Suppose this RH2 is one substance. Now if it will combine with oxygen, then it will give rise to R plus H2O2. This is hydrogen peroxide. Now this is carried out by the that peroxisome, and the enzyme is peroxidase. Now this hydrogen peroxide, it is quite toxic to the cell. So again it can break down the hydrogen peroxide. It can break down again by combining with suppose it will combine with again with the substrate, then it will give rise to R and two molecules of water, and here the enzyme is catalase. And both this peroxidase and catalase, both these enzymes are present in the peroxisome. That is why its name, because it is generating hydrogen peroxide and also breaking hydrogen peroxide in presence of these two enzymes. And particularly this catalase enzyme, this is known as marker enzyme of the peroxisome. This is known as marker enzyme, and it constitutes about 40% of the entire enzymes or proteins that are present inside the peroxisome. Now the peroxisome usually it contains around 50 it contains around 50 oxidative enzymes, and you can tell that after mitochondria, if there is any other organelle which contains more number of oxidative enzymes and which can directly utilize the atmospheric oxygen, it is the peroxisome. It contains. Therefore, it is thought that perhaps the peroxisome is a vestige of an ancient organelle, and it was present in the pro-eukaryotes before the evolution of the mitochondria. We know that mitochondria contains enzymes for oxidation, but it is thought that this peroxisome is present, it is a vestige of some oxidative organelle, and still it is continuing that oxygen. And after mitochondria, this is the only organelle which utilizes directly atmospheric oxygen. So for that reason, this is very important. And in addition to this peroxidase and catalase, there are also some other enzymes like urate oxidase, then you will get that is D-amino acid oxidase, then alpha hydroxy acid oxidase, and also beta hydroxy acid oxidase. So these are the different enzymes they are present. Now, in addition to this, the peroxisome also contains some proteins, special proteins, that is they are known as docking proteins. They contain certain docking proteins, and these docking proteins are known as usually peroxins. And the function of this peroxin is they obtain the different types of substances from endoplasmic reticulum. Usually, the peroxisome is associated with the endoplasmic reticulum and chloroplast and mitochondria. So this is about this peroxisome. Now it performs different types of functions, and the functions are different in case of animal cells and plant cells. So what are the different types of functions? Now one of the very important function is in animals, and particularly in liver cells, it detoxifies alcohol that we drink. So that is one of the very important function. In liver cells, it detoxifies alcohol that we drink. In addition to alcohol, also it can oxidize. It also oxidizes the nitrates and phenol compounds also. So this is one of the very important functions, particularly in case of livers. Another is there are certain compounds, that is very unusual compounds. We can call them as xenobiotics. So unusual substances, and we can call them as xenobiotics. Like one example, we can take that of the D-amino acid alkanes. Now this is D-amino acid alkanes. These are unusual substances. This cannot be utilized. This cannot be broken down by any compound in the cell, but it can be destroyed only by the peroxisome. This is very important. Then another important function is breaks long chain fatty acids in animal cells. Only in animal cells, not in plant cells. Now in case of plants, what is the function? In plants, one of the very important function is, in case of C3 plants, there is a special phenomenon known as photorespiration, which is not present in C4 plants. So in particular plants, in C3 plants, it receives glycolate from chloroplast, and then oxidizes it with by means of atmospheric oxygen to convert it to glyoxylate, and converts it to glyoxylate by using atmospheric oxygen. That means atmospheric oxygen is directly utilized. Then that glyoxylate will enter into the mitochondria, where it will be converted to the amino acid, that is glycine. And in addition to this important function, there is also, that is another function is there in roots. The fixed nitrogen is converted to urates, and the nitrogen compounds are transported in the plants in the form of urates. So these are the important functions assigned to the peroxisomes. Now let us discuss the second one. The second one is glyoxysomes. The glyoxysomes, they are considered as specialized peroxisomes and presented transiently only in germinating oily seeds like that of the castor or coconut, any oily seed. Because in oily seeds there are fatty substances there. So the different enzymes for breakdown of these long chain fatty acids are present in glyoxysomes, particularly in case of oily seeds, in case of plants. So the glyoxysomes, they are present only in case of plants. They are not present in case of animals. And these are specialized peroxisomes. These are specialized peroxisomes. Why specially peroxisomes? Because they appear in the germinating seeds. They break the fatty acids by means of a glyoxylate cycle. Then after the fat is completed, they disappear. They are converted to peroxisomes, and again they will reappear during senescence, because they will be again required for breakdown of the fats and mobilization of fats. That is why they are known as specialized peroxisomes, and they are mainly responsible for the beta oxidation of fatty acids through glyoxylate cycle. Cycle. So that is their importance. And again, after the fats are completed, they will disappear, and again they will reappear. So that is why they are present only in case of plants. So one thing is very important. In case of plants, the beta oxidation or the breakdown of fats takes place in case of glyoxysomes, while in case of animal cells, it takes place in peroxisomes. This should be very clear. So these two are the two microbodies, and from these two also sometimes questions are asked. I hope now your idea regarding the peroxisome, glyoxysome, and what is their between the similarity between them and the difference between them is clear to you. So thank you very much. Wish you a very bright future. God bless you.